So I would like first to thank EPNOE for the kind invitation. It's always a pleasure to meet some people from this organization. So, and also a pleasure to explain what we have been doing in our group using polysaccharides, and in particular polysaccharides from aquatic origin for biomedical application. And I will focus much more on regenerative medicine application. So for people that does not know where we are coming from, so University of Aveiro is in the coast, in the Atlantic coast, not far away from Porto. So we are north from Portugal. Is a quite pleasant city where our university is based. Is a quite new university with 47 years old, very strong in some areas, in particular materials, science and engineering. And so in CICECO, we have our research group where we are trying to combine cells and materials using different types of tools, using bioengineering tools, chemistry, some chemistry, nano and macro technologies. And then we try to develop new concept devices for both ex vivo or in vitro, as well as implantable applications. And in particular, we are very interested in developing new ideas for tissue engineering and cell therapies. And for that, we have been working with tissue engineering. I'm sure that most of you are aware about this emerging field in medicine, where, for example, if we consider autologous approaches, cells are isolated from the patient, expanded, and then they are combining with biomaterials. That's why polysaccharides and other types of biomaterials are very important to where we can process and use them to combine with cells as three-dimensional matrices, where using adequate biochemical and mechanical stimuli, for example, using bioreactors, we can really stimulate these cells that have been extracted from the patient to develop their own extracellular matrix, differentiate and originate functional tissues that will really be able to be implanted again into the patient. So depending on the tissue that we wish to engineering, besides the right choice of cells, we really need to select wisely the biomaterials to be used, and that will be integrated with cells at different length scales. So among the large choice of biomaterials, of course, natural polymers are very important. They constitute really a large portfolio of different types of structures that are, and this is an advantage, they are already engaged in the functioning of living creatures. And therefore, they are often biocompatible and sometimes they are biodegradable. Among them, polysaccharides, and I will focus, of course, only on this kind of class of materials, are a very important and chemically versatile class of biopolymers. We have been working with proteins and polyesters, but I will focus on polysaccharides. And in particular, we are interested in working with marine-derived biopolymers, and we have several projects on this topic, where the general idea is really to take the amazing, rich and sustainable resources from the marine environment, such as algae, crustaceans, sponges of jellyfish, to extract polysaccharides and proteins. And this is really important not only for our group, but in terms of strategy in Portugal, in terms of economy and research. Because if you look at our exclusive economic zone, this is really like 18 times bigger than our lands in terms of area. So it's a very big area that we can explore by different ways. And in fact, we are now negotiating. So the actual EEZ is in these regions, but now we are negotiating with the United Nations to more than double the area. And so the idea is to have something like more than the area of India to explore in terms of aquatic resources. So polysaccharides such as chitosan, alginate, or laminarin or laminaran are among the most abundant macromolecules that are synthesized in marine organisms, and therefore we can extract from the sea. So laminarin is very abundant, but it's surprisingly has received very little attention, despite it has potential as anti-inflammatory, antioxidant, and anti-tumoral or even anticoagulant material. So this kind of polysaccharide is a low molecular weight branch macromolecule, mainly used as energy storage glucan in brown algae. So the structure is composed by glucose units linked by these beta-1,3 glucosidic bonds, as well as some branches, one with the beta-1,6 bonds, which are responsible. So this kind of organization leads to a material with usually low viscosity, because also the molecular weight is not very high normally, non-cytotoxic, and also it's very, very soluble in water and even in some organic solvents. So we use this kind of very simple polysaccharides to show the potential with chemical modifications to obtain an extent range of properties that could lead to a new library of multifunctional macromolecules. So to develop a modular and flexible functionalization strategies that result in the incorporation of the different types of very, very defined functional groups into the laminarine backbone, we basically use two approaches. So first, an allele derivative was obtained via nucleophilic substitution process that involved the reaction of an hydroxyl group of laminarine with allelation agents, such as, for example, a lime bromide. And these allele groups can then react with thiol-bearing compounds using a thiol Michael additions that permits, for example, to incorporate groups such as carboxylic acid, amine groups, or even thiol groups into the laminarine. And another approach, so an aldehyde-bearing biopolymer can be obtained by the oxidation process of laminarine, and these oxidized macromolecules, it's quite useful because these aldehydes are much more, of course, reactive than the original hydroxyl groups, and we can obtain other materials, for example, using reductive amination. And we can use these kind of materials for different applications, either in the soluble form or by cross-linking by forming hydrogels. And hydrogels are really a very important class of biomaterials that can recapitulate many aspects of the mechanical, water content, and biochemical environment that exists in cellular microenvironment. So they can be prepared with a very defined architecture that can be designed to have, for example, a tuned interface with cells. It enables basically the encapsulation of any type of cells. Also, we can use as reservoirs of signaling proteins or therapeutic molecules that can also control cell behavior. And also, hydrogels are the essential ingredients for bioprinting or injectable hybrid devices. So using this kind of laminarine derivatives that I just mentioned, we can explore different ways to prepare hydrogels. And I'm just showing three possibilities that really shows the versatility of this kind of systems. So in the case of a chemical coupling, the laminarine with amine groups was combined with aldehyde polysaccharides, again, laminarine with after oxidation, to produce hydrogels via the formation of this kind of dynamic bonds that results from the reactions between these two groups. And these kind of hydrogels can be formed into physiological conditions in the time scales of minutes. So in the second strategy, the combination of thiol modified laminarine with this kind of alkane laminarine derivatives can undergo gelation, in this case by phytocross-linking. And this enables to originate hydrogels networks with controllable shapes, as you can see in here, able to sustain the encapsulation of cells. Again, this can be done in mild condition. And this is really because we can, this is mediated by a radical-based mechanisms promoted by the irradiation of an initiator, and we can use low-intensity UV or blue light to trigger this kind of process. And we can generate hydrogels in the time scales of seconds, which can be quite adequate, for example, to be integrated in bioprinting procedures. Finally, we also explored the possibility of cross-linking doppler derivatives through the coordination with ions. So this kind of catechol groups that also exist in marine environments, for example, in mussels to attach to rocks. So this is also quite a bio-inspired strategies to obtain hydrogels and adhesive materials. So the idea is, depending on the pH, we can have different degrees of coordination with iron, and so we can originate hydrogels with iron ions, and we can recover the liquid phase by using an alkylating agent such as EDTA. So another way to form very fast photocross-linkable laminarin-based precursors is through the direct reaction with glycidyl methacrylate. So we obtain what we call methacrylated laminarin, and this can be done with different extensions of modification. So we can generate, for example, low degree or high degree of modification. We can play with the concentration of the polymer in solutions to originate just by light and using adequate photoinitiators, hydrogels with tunable mechanical properties such as stiffness that will also dictate the different swelling degrees. And everything is very important in terms of biomedical applications, not only, for example, for the delivery of drugs, but also to encapsulate cells. And we know that cells react very strongly with the mechanical stiffness and the mechanical viscoelastic environment where they are embedded. So this kind of cell, any type of cells basically can be encapsulated in this kind of hydrogels. We could see that the cells could maintain their viability, provided that the nutrients can enter inside the hydrogels. And this is a very important topic, okay? So hydrogels, in fact, are fantastic matrices to accommodate cells, but in bulky hydrogels, they can have limited capability to diffuse oxygen, glucose, of course, and other nutrients. So that's why we recently developed a completely new concept where glucose is generated in situ by the own hydrogel degradation, so allowing cell survival and functions without any need for further entrance of glucose. So we will take the advantage that laminarin is structurally a polyglucose macromolecules, and is, as I said previously, is used as an energy reservoir for brown algae. So for this purpose, so photocross-linkable hydrogels, so you obtain with methacrylated laminarin, were fabricated, immobilizing adequate enzymes to provide glucose feeding for metabolic activity of incorporated cells through a controlled degradation of the own polysaccharide where the cell is embedded. So we preferentially use glucoamylase as the degradation agent because it is an exoenzyme allowing just the degradation of the non-reducing sugar end units of the polysaccharides, thus resulting in direct glucose production and continuous direct glucose productions without cleaving randomly the laminarin backbone and preserving, of course, the hydrogel structural integrity. So together with our colleagues at University of Aveiro, Professor Manuel Coimbra and Claudia Nunes, we could conclude that glucoamylase could provoke the release among time of glucose in the concentration-dependent manner in these three situations. So methacrylated laminarin in solutions together with solubilized glucoamylase, methacrylated laminarin hydrogels in the solution containing glucoamylase, and more interesting, hydrogels that already encapsulate glucoamylase inside. And you can see that with time you have a cumulative release of glucose. So what's happened when we include cells inside? So we encapsulated cells, in this case human mesenchymal stem cells inside the hydrogels that were put in different solutions with a normal medium and medium without glucose. And you can see that in those cases, even when the system has no glucose at all to feed the cells, if you have enzyme inside the hydrogels, they can really maintain the survival of cells because the enzyme will internally degrade and release the necessary glucose for cell survival. And you can see that without enzymes, the cells cannot survive, for example, after 14 days of culture. So what we could see is that the encapsulated stem cells could proliferate inside the hydrogels containing the enzyme. For example, you can see for 0.7% of the enzyme, if we quantify the DNA content, so we are really see if we have cell proliferation, we can see significant increase in DNA content, therefore in cell proliferation inside these elastic hydrogels. And this is not the usually results that we obtain when cells are encapsulated in elastic hydrogels. So we also evaluated the performance of these hydrogels in vivo. So the hydrogels were subcutaneously implanted in mice, and histological and live death characterization of the explants were carried out after 7 and 14 days. But also we did in vivo ultrasound tracking of the implanted laminarin hydrogels, and we could see that laminarin hydrogels maintain intact without any sign of degradation. However, in the case of 0.7% of enzyme inside the hydrogels, we could see a significant reduction in size, indicating that we really have further degradation, but the structural integrity of the hydrogels were maintained. So over 2 weeks of implantation, encapsulated cells, again MSC cells, demonstrated significant high viability compared with the no enzyme solution. So you can see that with 0.7% of enzymes, the cells implanted into the back of the mice could survive up to 14 days, while the systems without no enzymes, the cells would not survive more than 7 days. And so as a conclusion, these cell-degradable hydrogels can really serve not only as 3D scaffolds for structural purposes, but also they provide a sustainable nutrition sources through gradual release of glucose over time. And this is really a concept that was never explored before. So besides nutrition, it's very important to provide cell adhesive cues to hydrogels and the capability of these systems to be reconfigurable on demand. And the recent works have pushing the limits of hydrogels designed to introduce dynamic environments to cell. And we can distinguish a particular type of advanced hydrogels that are called chemodynamics. So basically, those are based on reversible chemical interactions that enable them to respond to biochemical or mechanochemical cues and adapt accordingly. So there are some examples in the literature, including the case of viscoelastic hydrogels that have stress relaxation properties, and therefore the cells can relax their networks under cell forces and allow the cells to move through the network. And this is something that happens also in native tissues, this kind of dynamic ability for cells to be accommodated. So we have been working on the development of hydrogel systems inspired by the fact that our tissues do not behave like static hydrogels, but actually they deform and accommodate mechanical stresses. So to do so, we took inspiration from the native extracellular matrix, for example, collagen fibers, where the cross-linking is mediated by lysyl oxidase on collagen fibrils. And it's basically based on the formation of dynamic bonds by the reactions between aldehyde and amine groups. So those are reversible linkage that can break and deform continuously, allowing for the network rearrangement. And so to reproduce these natural phenomena, we started by engineering laminarin polysaccharides with pendant aldehyde groups. And this was presented before in one of the first slides by oxidation. And then we combined this oxidized laminarin with gelatin that exhibits free amine groups, and therefore this kind of reactions can occur in physiological conditions. Okay, so of course we can play with the relation between the two biopolymers, and for example, if the laminarin don't contain aldehyde groups, so we are not going to form any hydrogels. And the hydrogels can be formed with different shapes depending on the molds that we are using. So we then hypothesized that these dynamic crosslinks could convert incoming mechanical inputs into architectural rearrangement. In fact, the hydrogels could be deformed and retain the new shape after a mechanical loading. As you can see, for example, from this image, that shows the consequence of an one direction compression leading to hydrogel deformation. And after taking out the plate, we can see that a new geometry is maintained. So you can see, and you can compare with the situation of native gelatin hydrogel, where the geometry or the piece is easily crumbled under the same condition. And so again, this is really because of the dynamic covalent bonds of the imine bonds that results from the aldehyde and amine groups reactions that can break by the action of the mechanical force and reform again, and therefore the network is stabilized under the new deformed shape, stabilizing also the microscopic object. So we leverage these interesting properties to install microtopographies on such kind of plastic-like hydrogels on demand. So you can see from the video and also from the SEM picture that the hydrogel display the new topographical features inherited in this case from the coin pattern upon the application of a mechanical load. Very simple. So also the hydrogels could retain their topographies for over at least 1 week in cell culture medium, which of course it's interesting because it opens a new way to instruct cells through patterns. So the student, which is Pedro, was saying that of course, unfortunately, the cells cannot read money. They do not know what to do with this kind of topographies. But of course, we can imprint other types of patterns to instruct cell behavior in fact. So what we did, so we used adequate molds. In this case, the hydrogels could be reconfigured with anisotropic nanopatterns like the ones that you are seeing. So you can see that we can see clearly this intercalated nano-ridge array originating from this imprinting process. And when we see human cells on the top of these hydrogels, the cells really sense the underlying nanotopography and could readily align along the pattern's directions, even when we are using high cell densities. So we can see from a fluorescent microscope that the corresponding radial plots and also the direct observations, we could see that really the cells could align in the directions on the nanopattern features as compared with the non-pattern control groups where the cells are completely random. So this we can clearly conclude from this work that we can really reprogram the topography and the surface geometry and this on hydrogels, on this kind of plastic hydrogels, and with that we could control cell mechanisms, including orientation. And we know, I don't have time for that, we know that this kind of alignment could also lead for example, stem cell differentiation, for example, into bone-like cells. So in the hydrogels that I just presented that have pseudoplastic behavior are really an exception, because usually hydrogels are quite elastic and also quite fragile, okay? And this is due to the high water content and also heterogeneity of sizes and organization of the chains between the cross-linking points. So by playing with the chemical and macromolecular design, several strategies have been proposed to increase the toughness and also the strength in hydrogels. And we resumed that in the review paper some years ago. And this is very important because for several biomedical applications, we really need to have strong hydrogels for the application. So in particular, one strategy corresponds to these double network strategies that involves the overlapping of a stronger cross-linking network that will provide stiffness, that is shown here as a blue network. And we combine this network with a more loosely cross-linking network, here schematized by the red network, that will be able to dissipate the energy of any crack propagation due to a preferable network breaking, maintaining the first network intact and therefore the first network can recover any change in geometry that is imposed. So as an example, we use a full chitosan-based hydrogel. Again, of course, a very important type of marine-derived polysaccharides. And we prepare these double networks that is entirely based on chitosan. So we combine a low molecular weight metacrylated chitosan that can cross-link with light. So this is the orange network that is our covalent cross-linking network. And we mix that with a medium molecular weight chitosan that was ionically cross-linking with 3-polyphosphate, which are the chains here in green. So to obtain the hydrogels, of course, were characterized by compression tests. And I will not going into, of course, into details, but just the hydrogel consisting in chemical cross-linking, so just with a covalent chitosan, we could see that the usual fragile behavior with a maximum stress of only 80 kilopascal, and it could collapse completely after more or less 50% of strain. Okay. So for the double network hydrogels, we observe a completely different type of behavior, although this hydrogel has a very similar molecular composition and also the same water content. And in this case, for the double network, basically the hydrogels did not break, at least up to 90% of strain. And it could recover, as you can see from the video, the geometry after being imposed with this compression. So moreover, it could reach the amazing value of 20 megapascal of strain, which at that time, this was the strongest hydrogel ever reported based on any type of natural polymers. Moreover, the advantage of this hydrogel is that they can be prepared from the liquid precursors using mild conditions. So we can encapsulate cells and the cells can maintain their viability, which is also, of course, very important in tissue engineering. So these examples of hydrogels that are shown, you saw that the geometries of the materials that were tested, for example, for mechanical tests, have a very simple geometry. But of course, for real application, we really need to accommodate the geometry to the defect size, for example. So, and this is important for tissue engineering. And that's why bioprinting procedures have been popularized, and they are very, very used nowadays in the field of tissue engineering. Usually they are based on, as you can see in this video, on layer by layer deposition of multiple extruded materials that can be stabilized and they can be transformed into hydrogels by different processes. So in the last year, some strategies have been proposed to bioprint more complex structures, but with completely freedom shape maintenance. In this case, usually people are using a supporting material that provides a 3D environment in which hydrogels inks can be extruded and deposited in gravity-defying spatial arrangement. So usually they are based on the use of beds or gem microgels through which the bioinks is extruded independently in the 3 orthogonal directions. And these allow basically to process any type of structures in the space. So in our group, we developed a pure continuous viscous support that allows the fixation of complex centimeter scale 3D constructs with completely arbitrary design. In this case, what you are seeing is the green liquid is an alginate solution that is instantaneously cross-linked by the calcium ion presented in the own supporting bath. Of course, we can extend this kind of alginate 3D structures, for example, using CAD models and processed using a bioprinting and using the same kind of supporting matrix. So this gives rise to, in this case, to a suspending object. And in this case, you can see that the line of gravity falls outside the structure support base, and therefore these kind of structures could never be prepared using these layer by layer bioprinting processes. Moreover, what we observe is that with this pseudoplastic support, we can really go for unprecedented printing velocities comparing with other embedded 3D printing methodologies that usually use hydrogel microbeads. So other structures can be obtained, such as you can see this asymmetric tree-like structure. The supporting bus is based on xanthan gum solution, and this solution exhibits a clear Thier-Schinning behavior. And this is very important. So these properties allows the solution to fluidize under the action of the motion of the needle, offering little resistance for the local extrusion of the bioinks inside this bath. After the passing of the needle, the solution acquires again a higher viscosity in the resting phase, and it will allow for the fixation of the previously deposited extruded. So you can easily extrude, and then what is extruded will be stabilized again. So we could see that there is a low mixing between the supporting bus and the filament that is extruded. Again, it's an alginate filament. And therefore, and moreover, the object that is bioprinted can be recovered easily because xanthan gum is water soluble, and just by washing with water or PBS, we can recover the objects that we wish. So we could also imprint together with cells. Everything is really can be done in physiological condition. As you can see, cells can maintain the viability. So this is really a very interesting way to extend the normal 3D printing of hydrogels to create very complex shapes. Again, we must be very careful because the cells can survive provided that the filaments and the structure have a very low distance between the cells and the surface so that the nutrients can enter. And again, this is a very important aspect that I already mentioned before in these self-feeding hydrogels with glucose. So so again, very important. So we can have problems in terms of excess of nutrients, especially when we have very large hydrogel constructs. And moreover, usually cells have very limited mobility because they are embedded in the quite elastic matrix. So recently, and we have been working a lot on trying to develop compartments for cells that are not hydrogels, but they are much more viscose or liquefied compartments. And this is really also bio-inspired, but you can see that many biological processes are taking place in liquefied compartments. For example, inside cell, lysozymes are liquefied vesicles where complex enzymatic processes are taking place. Even cells, we can consider that cells are really quite viscous environments where you have all the organelles inside. And if you go for more complex organisms, for example, in terms of development, we can recognize that embryos grow in liquefied compartments, such as in eggs, or even in placentas for even more complex organisms. So we have proposed the concept of liquefied capsules with adhesive sites for cells, because usually cells and the cells that we are using needs to have some kind of surface to attach. So they are really anchorage-dependent cells, otherwise they will not survive. And this kind of liquefied compartments are enveloped with a perm-selective membrane, allowing the entrance of nutrients and the exit of waste products. And the cells are completely free to explore the entire volume of the compartments because this is really liquid. So we can include any type of elements that we want inside. For example, particles where cells can attach. Again, very important, this kind of attachment motifs for cells, and other kinds of proteins if we want to provide some signals to the cells. So it's a very, very versatile kind of units that can be then integrated using bioprinting or other kinds of bottom-up strategies to prepare bigger elements. So this is just the basic units. So how we are preparing these units? So this is just the first generation processing. We are now working on the second one with one pot kind of processing. So the first one is based on the preparation of alginate beads. So everybody in the polysaccharides knows how to prepare alginate beads by spraying droplets of alginate solution in the calcium containing bath. But in this case, the alginate beads also contains, and the alginate precursor solution also includes cells and microparticles, all the elements that we want to see in the final capsules. So the beads of alginate are obtained, containing, immobilizing the cells and microparticles, and then we are using layer by layer technology to coat the beads using polysaccharides like alginate and chitosan. And in this case, we also use polylysine. So after having this shell form, and we can control the nature of the polymers, the number of layers. So this will dictate the final mechanical properties of the capsules and also the diffusion properties. After having the shell form, we used EDTA to collate the calcium ions, and this will result in the liquified capsules that will immobilize inside all the particles and cells that were previously in the initial beads. Of course, they will not go outside because we have the shell that is kept intact even after the collation process. So it's very important to know already from the very beginning what we want to put inside the capsules, what type of cells, the particles, because it will not be possible to take out or to put other things after everything is formed, okay? So, and we hope that everything is ready so that the micro tissues can be formed in a very autonomous way. So we can play, of course, with different processing parameters like flow rate, spray voltage to originate capsules that can be obtained with a very narrow distribution range in size, but we can play with these parameters to obtain capsules with different sizes that can go from 200 micrometers to 1 or more millimeters. So these capsules can be then incorporate whatever we want inside. So this is just a scheme where you see a capsule liquefied inside. You can see inside, for example, the incorporation of stem cells here in blue that can attach to these microparticles. We also can incorporate endothelial cells here in red that can release factors so that stem cells can differentiate into osteoblasts, which are the cells from bone. They will start to calcify, as you can see schematically. They can interact in between, particles can be glued together by the action of cells, and in the end we can form a micro tissue inside the capsules, okay? So the definition of the initial cell composition is really relevant, and I would like to show you an example where this can be seen. So if we, for example, encapsulate just stem cells, so those are stem cells from human origin, from adipose tissue, after 21 days in culture, you can see that the cells can maintain their viability because inside the capsules, you have the easy access of nutrients and oxygen, but you don't see any indication of mineralization. For example, alizarin red staining, you don't see any red, and you don't see any osteopontine staining, so you don't see any green in here. So the cells were kept alive, but they maintain their stemness. They are kept as stem cells. But when we combine stem cells with endothelial cells, also from human origin, and if we combine in the capsules, so we call them co-culture capsules. After 21 days, you can see massive signals of staining of mineralized material. So this is calcium phosphate and also osteopontine. So this clearly indicates that endothelial cells release signals such as BMP2 that trigger the differentiation of the stem cells into bone cells, and we start to have a mineralized tissue inside the capsules in a very autonomous way in vitro, without any osteogenic factor. So this is very important. So we went also for in vivo experiments, and you can see what happens after implanted subcutaneously in nude mice, these capsules with just stem cells and the co-culture subcutaneously. So after 6 weeks, the capsules were retrieved and they were analyzed. I will just show one slide with the results, okay? So you can see that the monocapsules the collagen was not almost no present, so the stem cells did not produce many extracellular matrix. No alizarin red staining could be seen, as well as almost no osteopontin staining. But in the co-culture capsules, you could see much more content of extracellular matrix that were generated by the cells inside the capsules. So those are histology or histochemical slices of the capsules after retrieved from the back of the animal. And also a lot of calcium phosphate. And this is very important because this calcium phosphate, this hydroxyapatite, was generated by the cells, the human cells, inside the capsules, because in subcutaneous implantation there is no bone around, and also a lot of osteopontin staining. Okay, so this gives a lot of possibilities to explore this kind of capsules as units that can be integrated in more complex devices to originate the type of tissue that we want. In this case, this was generating bone, but we have been working with cartilage, and we want to work with other type of tissues, and depending on the ecosystem that we include inside these polysaccharide bags, we can generate different type of tissues. So I think this was my last slide, so I'd like to thank everybody, but of course the summary. So I hope that I could show you that natural polymers, in particular polysaccharides, are very versatile. You can do a lot of chemistry modification. For example, we can have systems that can release glucose on demand by encapsulating adequate enzymes, and this could feed the cells that are inside. You can combine polysaccharides with other biopolymers such as gelatin or other proteins to have mechanoconfigured hydrogels. You can play with the macromolecular design to have systems with very high toughness, which is very important. We can also play with bioprinting, and we propose a new way to bioprint systems with out-of-plane possibilities, so to much more versatile. And finally, I could show another completely different type of ways to use polysaccharides to prepare this kind of artificial placenta, where we can produce microtissues using adequate cells inside. So finally, I'd like to thank all the collaborators, not only at University of Aveiro in Portugal, but all over the world. The funding, without funding, of course, this could not be done, in particularly from national agencies and from European grants. Of course, I always like to acknowledge the people that we have in the lab. Especially, they work very hard, especially in those difficult times. Again, as this was in the introduction, Materials Today Bio is a almost recent journal from Elsevier, from the very prestigious Materials Today family, and we are receiving manuscripts that should combine materials, any type of materials, with biology. That can be biomaterials, sensors, so whatever you can think. So those are some of the topics that are covered by the journal. And finally, I would like to thank you very much for your attention. Thank you.
Thank you, Professor Mano. Now the floor is open for questions, and please write in the chat. So we can see from the chat if you have any questions. But while we're waiting for the question from the audience, I have quite few, but I can start with one. Is I'm working with wood-based polysaccharides from trees. I have been asked very often that the biodegradability, because we know that they cannot be degraded in the human or animal bodies. And what about the marine-based polysaccharides? Do you have any opinion or your thought?
So the marine-derived polysaccharides are quite versatile. So there are some polysaccharides that are degradable in vivo. For example, chitosan is one possibility, but most of them are not. For example, alginate or laminoran, So they are not naturally, because in our body, we don't have the necessary enzymes, okay? But we can take as an advantage. Of course, for some application, you don't really need to have degradation. But if you immobilize the necessary enzymes, you can artificially and you can, in a tunable way, to degrade these systems that are naturally not degraded. And I show an example of how glucomylase could degrade, for example, laminin to generate in situ glucose. Okay, so this is an advantage that is not degraded, but if you put the necessary enzymes, it will be.
Yeah, thanks. Now we have a few questions here. If we look at the list in the chat, the first one is from Professor Tina Nupele. So thank you for the presentation. What are typical molecular weights for laminarans?
Okay, very good. So usually they are low. I would say 5 kilodalton, something like 5 to 10. So they are usually quite low molecular weights, polysaccharides.
Okay, so are they naturally low, or did you need
usually,
to hydrolyze?
No, they are naturally low.
Okay. Yeah.
We would like to have some material with high molecular weight, but it's very, at least, so we are not specialists in extracting polysaccharides.
Okay.
But if anybody would have some ideas, we would be happy to consider higher molecular weight laminaran.
Okay, very good. So the next question is from Xue Zhang. Thank you, Professor, for the nice talk. May I ask, do you use commercial laminaran or lab fabricated one?
Okay.
So,
yes, in this case, for these works, we use commercially available laminaran. I'm not sure if it's from Sigma, but it's commercially available.
Yeah, okay. The next question is from Willem.
No, this is not a question. Yes, I think Pedro can reply. So if this, the video can be then see later. I'm not sure.
Yeah, that's right. Yes. Okay. Yeah, then question from Pedro. I'm interested about the direction of the cell growth using microstructures. You mentioned nano roughness. In your opinion, how rough nano or micro roughness should the surface be?
Okay, very good question, Pedro. What we saw is that we can have cell orientation either using nano topography, nano patterns, or micro patterns. We are not really expert in mechanotransduction. I think the effects are different to what they are doing with cells, but in both cases, you can have cell orientation. In this case, we had something in the between nano and micro. But what we can see is that, and this some people have been working on that more in 2D, depending on this geometry of the patterns, you can have not only orientation, but also differentiation. And for that, the values of the distance between the ridges and the aids should be much more tuned in order to have cell differentiation. And we also work with that in pseudo 3D, not only in 2D, but in the space too. Okay, but I didn't have the time to present these works.
Yeah. Okay, and then the following question is from Xiaoju Wang. Thanks for your excellent presentation. I would like to know if the immobilization of glucoamylase enzyme may interfere the encapsulated cells in any aspects.
Yeah, so this, thank you very much. So of course, this is a very important. So as far as we just analyze the cell viability, okay, if the cells could proliferate, if they are viable. And what we conclude is that at least for the amounts of glucoamylase that we use, the cells did not, this was not compromising cell viability and cell survival. But of course, indeed, we didn't analyze, maybe it could have some effects in terms of genetic profile, but I'm not sure, okay? So this we didn't do, okay? Maybe they have some effect in some cell condition, but at least for what we saw, the cells could be kept alive.
Okay. Thank you. I see these are the questions so far. Are there more questions we can still take? Okay, and there's another question from Qingbo. Thank you for the wonderful presentation. May I ask, what is the rheology and mechanical property requirement for the supporting matrix for 3D printing? Yeah, that's very interesting.
Okay, yes. I cannot say you what are the values. So what we did was to you can check on the paper, at least for the rheology that we obtained, the conditions were enough. So you need to have something that should be viscous enough to sustain, even with the effect of gravity of the structures, okay? The density should be also similar. The density between the bath and the hydrogel, but this is more or less obvious. But also you need to have this thixotropic kind of behavior so that you can print without pushing the material. So the material should be able to open, so the viscosity should be able to decrease sufficiently low so that you can pass the needle. Okay? So we did some optimization and the results are in the paper that we published in Biofabrication, and you can look at that.
Yeah. Okay, then we have another question from Pedro. This placenta concept is interesting. Do you have the oxygen inside? Is there some mass transfer nutrients and oxygen through the walls or skins of the spheres?
There is. So and this we analyze. So we analyze the mass transport of oxygen and nutrients depending on the number of the layers in the shell. And so what we are using is the number of layers are not permits the passage of oxygen and glucose forever. Okay, so we can have cells for many, many weeks in normal culture medium. If the culture medium has oxygen and glucose, they will diffuse inside, and this we analyze. But now we are working in another ideas where we have self-oxygen generation. So you don't need oxygen from outside, which is much more interesting. And this is very important for some in vivo application where we implant devices where there is not so much oxygen available to maintain the survival of the cells.
Yeah, thank you. That's really inspiring. Okay, there's no more question. And maybe I can still ask the last question as a chair. There's an opportunity. So you had many different chemistry or chemical approaches, and you open the sugar ring by oxidation, and then there are two aldehydes. And did you notice if there's any impact on the biodegradability when you open the sugar ring, or have you looked into that, or would you think that would be a possibility?
I'm not sure. I'm sorry. I'm not sure if we did these degradation studies. You are saying just hydrolytic degradation, right?
Yeah.
So cleavage. I'm not sure if it will help the hydrolysis, the presence of aldehyde groups. Maybe there are more competent people than me to answer, like pure chemists. So we are just using chemistry as a users, okay, to prepare
for some biodegradability in the body. I mean, would that
in the body, I'm not sure, because I don't think that to have aldehydes, it will increase the degradation, enzymatic degradation. What will happen is the aldehydes can react with free amines in the body and fix the material, but in terms of degradation, I'm not sure if it will have an impact.
Yeah. Thank you.
I'm sorry.
Thank you, Professor Mano.
Thank you very much.
Thank you for coming to give us the plenary lecture. So if you have more questions, then just feel free to contact Professor João Mano. And thank you.